My name's John Saville. I've been lucky enough to be head of the College of Medicine, Veterinary Medicine for the last 12 years. Normally the principal introduces, I thought I'd lost our lecturer there.
Not that easy.
Normally the principal introduces inaugural lectures. Unfortunately he's been struck down by hopefully something self-limiting. So that pleasure falls to me this evening. There's a number of welcomes to be made, particularly to Malcolm's family. This must be the most nerve-wracking lecture he's ever given, because his wife, Professor Lindsay Thompson, is in the audience, and of course they did the alternative configuration not so long ago and are still together. So, so it's obviously a strong marriage. And welcome to Malcolm's mother, other relatives, and his sons Callum and Magnus. Also welcome to school students from Armadale Academy, Balerno High, Bathgate Academy, Boroughmuir, Craigmount, Falkirk, Firhill, James Gillespie, Lasswade, Linlithgow Academy, Lawnshill Academy, Musselburgh Grammar School, Newbattle, Pennacook, Portobello, Preston Lodge, St Augustine's, St Thomas of Aquins, the Royal High School, Tynecastle High and West Calder High. So welcome to all of you. And it's a great and special occasion this evening. Malcolm is one of ours. He graduated in medicine with an intercalated first-class honours quite a long time ago, I won't say when, and has really built his career in the Edinburgh area since then. He spent some time overseas in Australia as a research fellow at the National Stroke Research Institute, and as you'll discover, much of his own research has been in the very important area of stroke. He's a very distinguished colleague, and for example is now a member of the Committee for Human Medicines, which is a really important committee that signs off on new medicines for use at the Medicines and Healthcare Regulatory Agency, MHRA. So Malcolm's title is an interesting one. Rigour Mortis, How Bad Research is Killing Science, because I'd like to bet that his own research doesn't fall into the bad research category. I'm just going on a hunch here. So we're waiting with bated breath. Malcolm, over to you.
Wow. Well, thank you very much for those kind words of introduction, and thank you all for coming. It's a bit like throwing a party. You never quite know if you're going to have an empty room. So first of all, thank you for coming. A lot of my research is and was awful. Some of it I know why it was awful. Others I've yet to discover why. In the background to this slide, you can see, I hope, the names of many of the people that I've collaborated with over the years, and this is by way of an acknowledgement slide at the beginning, but also to tell you that the errors which follow are mine and not theirs. And in the corner here, I'd also like from the start to thank NHS Forth Valley for their great support over an association with them that goes back, I think, 20 years from when I was a medical registrar. They've been very supportive, and without that support and NHS working together with the universities, none of this would have been possible. And before I go very much further and forget, I would like to thank Lindsay and Callum and Magnus for their great support over the year. This is Lindsay's inaugural lecture in November last year. Those of you who know us will know that we're not at all competitive. But my own view is that once you adjust for age and comorbidity, we have reached this point at about an equal stage in life. So what are the purposes then of an inaugural lecture? Because you're faced with this thing which is completely unlike anything else you've ever been asked to do, and working out what it is that you're supposed to do is quite difficult. So I think there's maybe three things. First of all, to try to explain to family and friends what it is that you've actually been doing for all of these years. Secondly, to try and convince your colleagues, who always come along, that what you've been doing has actually had some utility, been of some use, and probably most importantly, to try and encourage the next generations of researchers and teachers to do the same sort of thing that you've been doing, but to do it better, to improve on what you've done. And what I'm going to try and do today is take you through what I've been doing to give you a flavour under each of those heading. So those of you who've read the title of my chair is that I'm a chair of translational neuroscience. So what on earth is translational neuroscience? Translational medicine for many years has been a buzzword that goes in grant applications because people like giving money at translational medicine. And there's probably as many different definitions of translational medicine as there are practitioners doing it. But my definition really simply is using information from one research domain, in my case, animal models of neurological diseases such as stroke, and applying those findings in another research domain, in my case, clinical trials in humans where we try and see if those treatments improve outcome. So this is an example of a translational road scientist, not a neuroscientist. This is from Wales, where they have a habit of writing road signs in Welsh as well as in English, presumably to promote their tourist industry. And what it says is no entry for heavy goods vehicles, residential site only. And when you look at what the Welsh actually says, it says, I'm not in the office at the moment, please send any work to be translated. So this translational road scientist, road signist rather, has sent his data into a black box and has believed without critique what's come out of the black box and has ended up making rather an idiot of themselves. So here's an example closer to home. This is an experimental technique called functional MRI scanning. And this is something which has been tested and used in hundreds of grant applications and spending millions of pounds of research income. And it's predicated on the basis that if you use a bit of your brain, it requires more energy. And if it requires more energy, the blood flow goes up. And if the blood flow goes up, you can detect that in a scanner. So you put people in a scanner, you get them to do a task, and then you do the scan to see which bit of the brain lights up. Now, these scanners are temperamental beasts, and they need to be set up and trimmed and modified all the time. And this group based in Boston had a competition to see who could bring in the most interesting piece of organic material to set up their scanner every Monday morning. And Craig Bennett every Monday morning walked past the fish market. And so he said, no one's had a salmon in the scanner before. So he got one mature Atlantic salmon to put in the scanner to set it up, to twiddle the knobs to get it working. And the salmon was about 18 inches long, weighed 3.8 pounds and wasn't alive at the time of the scanning. They didn't know whether the salmon was male or female, but they didn't think that was important given that it was dead. And once they'd set up the scan, I don't know what happened. I don't know if some volunteer didn't turn up that day or something, but they were left with scanning time with no one to scan. So they said, I'll tell you what, let's put this salmon through this test. So they administered to the salmon a task. This was an open-ended mentalizing task. And the salmon was shown a series of pictures of human subjects with a specified emotional valence. The people were happy or sad or grumpy or perplexed or wondering quite where the inaugural lecturer was going to go next. And they were asked to determine, the salmon was asked to determine which emotion the individual in the photograph must have been experiencing. And several active areas here in red in the salmon's brain cavity lit up. Remarkably, in just about the same area as almost always lights up when you do this experiment in humans. And so what they concluded was either we've stumbled onto a rather amazing discovery in terms of post-mortem ichthyological cognition. That's to say that dead salmon can think, or there's something wrong with this translational black box and how you interpret it. And they won the Ig Nobel Prize for Neuroscience in 2012, and rightly so, I say. So a bit of background to stroke for those non-medical in the audience. It's caused by an interruption, 4/5 caused by an interruption to the blood supply of part of the brain, and it stops that part of the brain from working. And the consequences of that to the person who gets a stroke depends on which part of the brain is affected. In a warning stroke, what we call a TIA, the blockage is temporary, and the neurological deficit, the weakness or whatever it is temporary. But if the blockage is permanent, that part of the brain dies, and perhaps other parts of the brain can take over some of that function and you get some recovery, perhaps not. And there's a couple of important things. I don't know if there's any cardiologists in. This is what a heart looks like to a neuroscientist. It takes me back. This is a little roundabout, a circle of blood, of a blood channel at the bottom of the brain called the circle of Willis. And the blood supply to the heart, actually from the heart to the circle of Willis, there's 4 different ways that blood can get there. And then from the circle of Willis, you end up with what's called end arteries, that there's only one way blood can get to this bit of brain, and that's through that artery. So if you block an artery in the neck, maybe that's not such a bad thing, but if you block an artery higher up, then you get a stroke. And so This is an eyesight test, I'm afraid. If you get a bit of crud in the heart, like you can get if your heartbeat is irregular, then that can be pumped through the heart and out of the system, and you see it moves up, and it gets to block a bit of the brain, a bit of the blood supply, and then that part of the brain gets damaged. Brain is a bit like my appetite for chocolate biscuits in that it has no short-term energy stores, and certainly on a Friday morning I've got no short-term energy stores, and if your brain doesn't get a continual supply of energy, then that part of the brain dies off. And the burden of stroke, about 5.5 million people dying worldwide every year from stroke, 1 every 6 seconds, about 60 since I started talking. 40% of them die, 30% of them becoming disabled as a result of their stroke. So a substantial challenge for medical research and for doctors to try and find treatments that might work. How have we got on trying to develop treatments for stroke? Well, the answer is not very well. When I was in Australia, we were interested in seeing whether there were treatments that we knew of, that we knew to be safe, that we could potentially give to patients in the back of an ambulance soon after they'd had their stroke, because we understand that the sooner you can give the treatment, potentially the more effective it would be. So we were looking for something like aspirin or antibiotics that we were already knew were safe in the stroke population, that we wouldn't have to do extensive scanning or whatever before we could give them their treatment. And we found over 1,000 treatments which had been reported that their effectiveness had been tested in experiments in stroke, either in cell culture or in tissue slices or in whole animals. And of these, over 600 had been tested in an animal model of stroke. And what happens in an animal model of stroke is that blood vessel that I show you getting blocked off, you block off a similar blood vessel in an animal, and then you see what happens to the animal, and some of the animals get the drug that you're interested in, and some of them don't. And then that tells you whether the drug has helped improve outcomes. So 603 tested in focal ischemia, remarkably almost 400 improving outcome in those animal models of stroke. So you would think that would give you quite a lot to be going on with as someone interested in doing clinical trials. And in fact, over 100 had been tested in clinical trials, about 18 tested in clinical trials never having been near an animal experiment, and 97 that had been tested through animals. And we're left, this was 10 years ago, remember, with 4 interventions, drugs, which improve outcome in stroke. And they're clot-busting treatment with TPA, and then stroke unit care and an operation to release the pressure inside the brain, and aspirin as things not really tested through animals. And although TPA had been tested in animals and does improve outcome in animals, the reason why people tested it in stroke wasn't because of that, it was because it worked in heart attacks, which is a similar disease but affecting a different organ. So why is it that we've got this, what we call an attrition rate, where only 1 in 1,000 of these interventions seems to have worked? Well, this is what one of those experiments might look like. What you do is you get a group of animals that don't get any drug, you give them a stroke, and you measure how big, this is cubic millimeters, the stroke has turned out to be in that group of animals. And then you can give them half dose of a drug and full dose of the drug, and this experiment is testing low-dose glutamate and homeopathic doses of arnica montana, reported in a journal called Perfusion. But when I tell you that half dose low-dose glutamate is 10 to the minus 120 molar, and full dose, low dose glutamate is 10 to the minus 60 molar, then presumably the only people in the audience who remember what Avogadro's constant is are the school visitors that we heard about earlier on. But 1 mole contains 6 times 10 to the 23 molecules, so there aren't any molecules of anything in either of these, which means either homeopathy is alive and well and living in the Uniformed Services Division at Bethesda, or there's something wrong with this experimental technique that we're using. And the other thing, some of you may notice, the other thing is the homeopaths have their axes, have their columns the wrong way round, because of course 10 to the minus 120 molar should be the more powerful treatment if we believe the percept of homeopathy. So they're wrong on 2 counts. So what could be going wrong with, and that's taking the mick a little bit, because I could show you any number of proper experiments using proper drugs, which came to equally erroneous conclusions. So what could be going wrong with this experimental approach? Well, we know, and have known now for some years, that in clinical trials there are things that we can do in the way that we design the trial that protects the study from risk of bias. And 2 of the most important things are randomization and blinding. And by randomization, we mean that when people go into the study, at that starting point, they should have an equal chance of either going into the treatment group, where you're testing the new drug, or the control group, which is the comparator. So if you lot were experimental animals for me, and I took this ermine group in the front here and put them in the control group, and the rest of you in the treatment group, then there's a chance that you'd be different for reasons other than the treatment I was giving you. You know, this group might be slightly cleverer or not. They might be slightly older or not, but they'd be different in a number of ways other than the drug of test. So it's important that the group allocation is done randomly. And blinding means that you do the experiment as much as possible without knowledge of which group the animal belongs to, which group the human belongs to, both when you're doing the experiment and when you measure the outcome. Now, Magnus. For many years, we seemed to have dinner table conversations about who would win a fight between a pigeon and a squirrel. And firm views were held, I think it's fair to say, on both sides of this argument, some advocating the prowess of the pigeon in such martial comings together, and some saying the squirrel was a dead cert to win. Now, of course, if you wanted to bring some evidence to bear, short of collecting a large numbers of pigeons and squirrels and seeing who did win, which would probably not be ethical, you could try and measure the martial prowess of each of these, for instance, by their body size, by the strength of their claws, by their latent aggression and tendency to violence. But if you were to do that and you firmly held a view about which was superior, then your views would be biased, because Magnus, of course, would say the squirrel was a clear leader. You know, clearly, we had this conversation last night. There's no point in even doing the research, and if you do the research, he's only going to get one answer. So what you should do is you should hide from the investigator the identity of the squirrel and the pigeon, and try and do as many of the assessments as you can, blinded to which is which. And if you imagine, instead of putting a pigeon in one box and a squirrel in the other, I'd put one rat that had had my go faster experimental treatment in this group, and the rat that hadn't had the treatment in this group. You could see why that might work. And this is an example, this is a real example of someone who looked over 50 years ago at this effect. So this is a guy called Ronnie Rosenthal, who is an experimental psychologist living in the west coast of the States, and he used to run courses in animal psychology for graduate students in the States. And at the end of their course, they would have a practical. And the practical that they had to do was to test cognition in rats. And they were testing this in a thing called a T maze, where the rat is put in at the stem of the T here, and then has to choose whether to move to the dark arm here or to the bright arm here. And they alternated which was dark and which was bright at random, and the dark arm was always reinforced with food. So the animals were being trained to turn to the dark side, if you like, where they would be rewarded with food. And the students had to test two groups of animals, maize-bright animals and maize-dull animals. Maize-bright animals bred for many generations at the University of California, Berkeley, which had got an extreme interest in animal intelligence, to be smart at acquiring knowledge and learning knowledge in mazes like this, and their country cousins who'd been bred and presumably inbred for many generations to be not quite so sharp. And what the students found was that the maize-bright animals were better from the start compared with their maize-dull colleagues, and they improved more during the course of the training. So they were smarter and they learned better than their maize-dull colleagues. And when he asked the students later on what they thought of their animals, they thought that these maize-bright animals were fluffy and friendly and inquisitive, and the sort of animal you'd want to take home and have as a pet. And these maize-dull animals tended to try and bite them. And of course, the experiment that he'd done wasn't an experiment on the rats. The experiment he'd done was an experiment on the students, because these two groups of animals were drawn at random from the same cages in the same animal house on the same morning at the start of this experiment. And the only difference between them was in the minds of the people doing the experiment, and presumably their behavior and their approach to the animals was slightly different, which is why some of them performed well in the maze and others tried to bite them. So let's look at drugs tested in stroke models then. This is a drug called NXY059, test marketed by AstraZeneca in the news recently under the trade name Cerevive, because they thought it would make your cerebrum survive. And as well as these animal data, there were 2 clinical trials involving 5,000 patients. And you can see here that in the high quality studies, where they randomized the studies, where they blinded the scientists during the study, and when they blinded the assessment of outcome, NXY059 was substantially and significantly less effective. And none of these 11 publications met all 3 of these quality criteria, and the 2 used by Astra to persuade doctors to persuade patients to join their study met none of these quality criteria. So you might wonder why a company like Astra is spending all its time and effort doing or promoting research which isn't great. The results of the SAINT-2 trial were released in October 2006, and this is what happened to the AstraZeneca share price on the day that those clinical trials results were published. And those of you who've been following the recent Pfizer takeover bid will have heard the guy from Astra saying that they were undervaluing the company because of the value of these drugs that were in the pipeline that were going to give them profits in future years, but they just hadn't got there quite yet. And this is what NXY was doing, lifting up the AstraZeneca share price because they thought it would work. And because we know how many shares there were in circulation and we know how much it dropped by, we can say that the industry at that time valued a successful neuroprotective drug at something like $2.8 billion, somewhere between the gross domestic product of Fiji and Barbados. So there are real powerful sources of bias influencing the way that we do our science, even if we think we're living in an isolated community. And the same thing happens for other diseases. Here's stroke, which you've seen. Here's animal models of multiple sclerosis, blinded studies, less efficacy. Here's Alzheimer's disease. Here's Parkinson's disease. So wherever we look in the neurosciences at least, we find that measures to reduce the risk of bias when they're not taken, these experiments overstate how good the drug is, and you could see how that might encourage you to take a drug to clinical trial when the evidence behind it isn't great. So how common is it that this happens? Well, unfortunately, it's far too common. These are the rates of randomization over about 500 stroke studies using animals, only 37% randomized. Only 37% blinded, only 3% with a power calculation. A power calculation is when you set out at the beginning of the experiment to work out how big it needs to be to find what you're looking for. And unless you do that up front, it imperils the statistical validity of what you're doing. So stroke is pretty poor here. And when we started producing this work about 10 years ago, people said, well, that's why you don't have treatments for stroke. It's because all you stroke researchers are lousy. You're awful. You don't do proper experiments. If only you were like the rest of us in the neurosciences who are much, much better. So we looked at the rest of the neurosciences, and unfortunately, they're not much, much better. And you can imagine what happened next is they said, well, that's why you don't have any good treatments for things like Alzheimer's disease and Parkinson's disease. It's because neuroscientists aren't very good at doing science. If only you were like the cancer doctors or the HIV doctors, then you would be getting decent results. Some of you may remember Al Gore, the greatest man who was never president of the United States because of all those hanging chads. But his major impressive contribution was this thing called Pubmed. The United States National Library of Medicine kept an index of every scientific publication relating to medicine ever. And Al Gore says, well, we've got the information. Lots of people could use it. Why don't we just make it freely available to everyone? Which is what he did. And so Pubmed now has something like 25 million records of scientific papers being added to at the rate of about 3,000 a day. So it's a huge repository, difficult to know what to make of it all. And each publication has got a unique number. And because it's got a unique number, if I select a random number between 1 and 25 million and go to Pubmed, I can fish out a randomly selected publication. Like if I say C13, I'll randomly select someone from the audience here. And what we did was we looked at 2,000 randomly selected publications, and we found that almost 300 of them reported experiments in the laboratory involving biological materials, cell culture, animal tissue slice, that sort of thing. And we looked across 5 different time periods of the reporting of blinding, in green here, creeping up to about 5%. Conflict of interest statement, doing quite well in recent years, and randomization creeping up perhaps to 30% when you take in vivo studies studying involving animals and in vitro studies studying involving cell culture and the like together. So you can imagine what people said next. They said, oh, but that's just the average. Of course, if you were to look in work that came from high-profile, high-quality institutions, then you would know that it was good. And scientists know who to trust and they know who they don't trust. So there used to be a thing called the Research Assessment Exercise 2008. And some of you will remember the Research Assessment Exercise 2008. It set out on behalf of the government to measure the quality of research produced by UK higher education institutions in order to guide funding decisions for the future. And they wrote a commentary on what they thought the state was of UK research at the time. They said, an outstanding contribution to the internationally excellent position of the UK in biomedical science and clinical and translational research. They said they were impressed by the strength within the basic neurosciences that were returned. And so when we read this, it didn't really fit with what we were seeing in our systematic reviews. So what we did was we looked at the 5 leading universities identified by RAE 2008 as being the top 5 institutions for life sciences research in the UK. And I'm pleased to say that Edinburgh is a member of that group, although I may be less pleased in a minute. And what we did, we looked at all of their research output in 2009 and 2010 that described experiments involving animals. So that was over 1,000 publications where we got 2 people to go and say, was this a randomized study? Was this a blinded study? What does it look like? And this is what it looks like. So randomization at about 15%, 2009, 2010, blinding less than 20%, inclusion-exclusion criteria about 10%, sample size calculation hardly at all. Now, I've not named these institutions because that would be unfair, but what you can see is that some institutions do particularly badly or particularly well on some of these measures, and I think this might be a more reasonable way of assessing the quality and the rigour of what universities produce than simply having folks sat around a table reading things and seeing what they think. And remember the last slide, the average from Pubmed? Well, the average in the years from 2006 onwards for reporting randomisation in a random selection from Pubmed is 36%. So the UK's 5 leading institutions produce research which is of lower rigour, which is less robust than if I'd just gone and plucked a random study out of Pubmed. And I think that is a cause for concern. It's such a sufficient cause for concern that this research assessment exercise is being repeated this year in something called the Research Excellence Framework, which is trying to do the same thing. Submissions went in November, results expected in December of this year. And so we made a Freedom of Information request to the people doing REF 2014 to say, can we see what you've got? And we can do this assessment. So when you say something like this, we can say, well, actually, you might like to moderate your effusive praise. And unfortunately, and very disappointingly, they said that they did not consider it was in the public interest for them to make that information available to us. Now, it may not be in their interests, but I think it sure as heck is in the public interest that we're able to try and moderate some of the more extreme claims that are made. And of course, then the next argument is, well, if you look in the journals of high impact, the journals that we all recognize being fantastic, then there's not a problem. Now, we measure how good is such a strong word. We measure journals' productivity by how often an article, for instance, that I've written in a journal is used by other scientists to support the case or to argue with the case they've made. How many times someone else has cited my work on average over the whole of a journal. And these are 2,500 publications in stroke, and this is the reporting of conflict of interest statements by deciles of journal impact factor. So this is the top 10% of journals by impact factor, and this is the bottom 10% of journals by impact factor. So high impact factor journals are good at asking for a conflict of interest statement. No journal is good at asking for a sample size calculation. Journals are reasonably good at asking people to say whether or not you blinded your experiments, but importantly, there is an inverse and a statistically significant inverse relationship between the reporting of randomization and the impact factor of the journal in which the work was published. So really, when Jonathan Seckle and others had come to me saying, what have you published so we can submit it to REF, I should have said, great news, guys. All of my work is published in these journals that nobody ever reads or nobody ever cites, and that means it's more likely to be robust, more likely to be methodologically sound. I want briefly to talk about 2 other issues about reporting, and I hesitate to say very much about the Morris water maze because it's named after Richard Morris, who's sitting in the second row there, who described it some years ago. But it's a test that's commonly used in neuroscience to assess learning and memory. And essentially it's a round pool of water with visual cues round about. I'm hoping that someone in the audience will correct me if I'm wrong. And there's a platform which initially has a little marker on it. So the animal is put in the water, and it sees the marker, swims to the marker, and then gets out of the water, so it's escaped, so it's happier. And then what you do is you take away the marker, but the platform is just underneath the water surface. So if by the visual cues it can remember whereabouts it's supposed to be, it can still swim there and get out. And animals that are good at that are better at learning and memory. Very simplistic. And these are experiments testing drugs and models of Alzheimer's disease. And what you can see, this is the size of the pool here, ranging from about 85 centimetres at the bottom to about 2 metres at the top. But almost a quarter of the studies don't actually tell you how big the pool is, that is, how far the animal had to swim. One of the main things that drives the animal out of the water is the water temperature, because if it's cold, they want to get out quickly, and if it's Portobello on a good day at 28 here, they might want to hang around in it for a while. And you can see that over 40% of the papers did not say how warm the water was, and the animals get trained, as I said, before being tested. And this is the number of training trials per day and the number of days training. And by far the largest group are those experiments that tell you neither. So trying to go and replicate this research is impossible because it's not described adequately. And there's another important issue about reporting dropouts. This is an experiment in stroke that we did comparing minocycline, an antibiotic, with control. And this is an experiment in 13 animals. And this is their behavioral outcome. Green is good and red is bad. And you can see, looking at this, that there seems to be more good up here. And these lines, you can draw lines which show that the control group are shifted up in that direction. And if you look at the median outcome, it's better in the minocycline group than the control group. But this isn't what we published. What we published, unusually, was this, that actually 5 animals in the minocycline group had died. And it's convention in focal ischemia, at least in the focal ischemia experiments that we've looked at, to ignore these animals. Just to pretend somehow that they didn't exist or that they never actually happened, or not to report that there were 5 deaths in that group. And you can see that when you account for that, first of all, you want to know this drug kills animals, but secondly, there's no behavioural difference between the two. I want to talk a little bit about publication bias. Publication bias is what happens when what goes from your laboratory notebooks into draft manuscripts then gets sent off to journals or not for consideration for publication. And some of them end up in journals, which is good, and some of them end up in the investigators' files because they tried once or twice, or it's a bit boring, or they're not particularly interested and they just give up on it. Now, if these groups of publications are different in some way from these groups of publications, then looking at the published literature is going to give you a distorted view of biological truth, because you're only going to be basing that judgment on a proportion of the experiments that have been done. And so we've been interested in publication bias, and this is in animal models of disease where the outcome that you're interested in is an improvement in outcome. So we give this animal a drug, it improves outcome, way hey, let's go to a clinical trial, isn't that fab? And we observe overall a 40% improvement in outcome. But when we use statistical techniques to correct for publication bias, that improvement in outcome shrinks by 10% to a 30% improvement in outcome. So that's worrying. What's really neat, but also worrying, is if you look at disease models where you're interested in harm. So if I want to put a new chemical onto the American market, I have to get approval from the Environmental Protection Agency, and one of the things that they're interested in is the reproductive toxicity of that chemical, whether it affects the health of pregnant mothers and their developing children. And the way that that's assessed is the companies pay people to do research where they give pregnant dams, pregnant female rats, the drug, and then they measure how the birth weight, how big the pups born to that litter are. And you can see that the observed harm is 0.3 units here, but when you correct for publication bias, the actual harm is about twice as much. So publication bias is a double-edged sword that moves in the direction of the interests of the people who are promoting and funding the research, and it's something that we really need to be aware of. So how big a hole does this make in published research? This is, if you like, the research potential. This is 100% research potential. If we take into account the risk of bias, we lose about 20%, makes a 20% hole in the middle. If we take into account publication bias, we lose about another 20%. And if we take into account people doing underpowered experiments, that's to say doing experiments that are too small actually reliably to find anything out at all, then we lose another 20-odd%. So overall, we lose perhaps 50 to 60% of the potential utility of the research that is done, 50 to 60% of research assistant time, of resources, of laboratory time, of chemicals, of laboratory animals, for 3 simple things that we could fix tomorrow if we chose to do. So, as someone who wants to do clinical trials in stroke, how on earth am I going to pick something that's worth taking to clinical trial if everything that precedes it in animal research is potentially misleading? Well, you can do a systematic review and a meta-analysis to ask how powerful the treatment really is, the quality of evidence, the range of evidence, whether there's evidence of a publication bias, and what the conditions are for maximum efficacy. So here's hypothermia for stroke, brain cooling for stroke. Seems to work in patients who've had brain injury due to cardiac arrest. There's lots of stories around about individuals who should have extensive brain injury but don't. My favourite was a man who came into my neurology clinic who'd fallen through the ice in a loch outside Edinburgh to try and rescue his dog. Not a smart thing to do. He'd been underwater for 3 hours. He was taken to Royal Infirmary, where 2 hours later, so he's been dead for 5 hours essentially, they opened his sternum, ripped it open, and they gently caressed his heart back to life. About 3 months later, he got out of ITU, and about a year later, he got back to work. So a remarkable outcome. The reason he was seeing me in the clinic was he'd got a bit of numbness down the side of his hands because they'd injured the nerves in his neck. So he told me a story, and then we both agreed that actually not bad outcome, really, and he went along his way. Many laboratories use cooling as a positive control in their animal studies. That's to say, when they're training someone up to do animal studies, they say, see if you can get hypothermia to work, because if you can't get hypothermia to work, you're probably not doing it right. And there's preliminary evidence from clinical trials in stroke that is encouraging. Peter Andrews in the audience is doing clinical trials in head injury, and there are existing clinical trials in cardiac arrest. So here's all of the animal data for hypothermia that we could find in 2007 with Bart van der Vorp, 100 publications, 200 experiments, over 3,000 animals, and this is each one of those individual experiments with an estimate of the uncertainty of this, but this is the overall estimate of how good it is, a 40% improvement in outcome. Now, if I was really keen on hypothermia, I'd quote all of these studies in my grant applications, and if I was the reviewer who wanted to knock back the study, I'd quote all of these saying you're talking rubbish. But if you've got a systematic review like this where you can see all of the data, then it gives you much more confidence in the overall estimate. So EuroHype 1 is a clinical trial that's powered to detect a 7% improvement in outcome. There's more than a 40% improvement in outcome in animals. In animals, the efficacy is maintained in high-quality studies, which is important. There is evidence of publication bias, but it still improves outcome by a third. There's a good range of evidence in animals for duration of cooling, delay to treatment, the intensity of cooling, animals with high blood pressure, and that allows us to test it in humans with a wide range of different kinds of stroke at different ages of different severities. And the conditions of maximum efficacy in animals is temperature dependent, and that allows us to pick a target temperature which is the sort of thing that we could achieve in the sort of place where these stroke patients are usually looked after. So you could take stroke patients to the intensive care unit and close them and cool them down to 33 degrees, but with the number of stroke patients that we have in the UK, there simply aren't enough ICU beds to make that a meaningful or feasible treatment. So this trial is testing whether a more modest cooling can deliver benefit. 11 patients in, nails being chewed, hoping for the best. And then are there other ways that we can make more use of animal data? Well, we've been advocating the use of multicenter animal studies to try and bring the same rigor that we have in clinical science towards animal science. So clinical trials are usually multicenter. They involve large numbers of animals. They've got considerable ethical review and monitoring of progress as the study goes on, and oversight of how the study is conducted in each center, things that animal studies simply don't have. If you've got enough animal studies, they're usually very good predictors of what happens in humans. This graph shows the delay in effectiveness over time of the clot-busting treatment TPA in animals, the first 90 minutes, the second 90 minutes. This is the same thing in humans, and Joanna's systematic review of human clinical trials, the delay in efficacy every 90 minutes. When I tell you that these data here involve something like 3,000 animals, then that tells you the sort of data sets that you need to be able to answer these questions. And this shows it again. This is the TPA data. As you start off with one experiment and add more and more and more and more and more experiments, you get a more precise estimate of how good the treatment is. NXY we talked about was taken to clinical trial when the evidence base was way up here. Hypothermia has got an evidence base way down here, simply in the terms of the number of animals that are involved. And in order to try and get to data sets of 1,000 or 2,000 or 3,000 animals, that would take one group many, many years to chew through those experiments. So with Emily Sena and with others in Australia and Germany and France and elsewhere, we are working on developing a framework for multicenter animal studies, putting together the different components, the same things that you'd have for a clinical trial, but putting them together for an animal study, not with the idea that every animal study would be a multicenter study. That would be daft. But if I'm going to try and persuade someone to spend 10, 20, 30 million euros on a clinical trial testing whether this drug works in humans, it might be worth spending £1 or £2 million making sure that it actually works in animals, given what we know about other drugs that have gone before. So what have we found out so far in this 10-year, 15-year journey? Well, animal studies which don't report simple measures to avoid bias give larger estimates for how good drugs are. Unfortunately, most animal studies don't report those simple measures which might reduce the risk of bias. Publication bias is important and it's prevalent. You can't assume that just because you read it in Nature it's true, and you can only find out these things by studying very large numbers of experiments and by assimilating the data and analysing the data. What are we looking into now? Well, can we advise scientists on more efficient research design? So we've got a project with Gillian Curry and Andrew Rice down at the London Pain Consortium, trying to work out if I'm trying to test a pain drug, is it better to do a small study and quite a severe experiment involving a small number of animals, or a much larger study where the stimulus is less severe? What's the different value of information that we get out of these different approaches? Important questions, not just for animal ethics, but also for improving research design. Can we use this approach? I was going to say I was getting into bed with my wife, but you know what I mean. Can we use this approach better to understand models of mental illness, a large unmet need in terms of the quality of animal research? Can we automate some of the techniques required? The first stage of this mental illness review, just sifting through the 14,000 things we get out of Pubmed that might be relevant, is taking 2 people 6 months to plough through. Can we teach a machine to do that? That would be great. Can we help publishers improve quality? We're working both with Nature and with the Public Library of Science journals to see if there's ways in which they can change their editorial policies to improve the quality of publication. 3,000 publications a day new to Pubmed. How on earth is anyone going to keep up? How on earth? And can we build tools, particularly automated tools, that might allow us to be a bit better at catching up with what is already known? Currently, most of these 3,000 publications go into little silos, and that work is known only to a handful, perhaps 100 people if you're lucky. How can we make that information more widely available in a digestible way without people having to read 3,000 publications a day? And finally, can we use this approach to design better clinical trials? So now the good news. These are deaths from stroke in Scotland for patients between the age of 70 and 79. Starting from 1950, when the death rate was about 1,600 per 100,000 population per year, and falling precipitously as a skier off a mountain to about 400 per year just now. And that's in spite of us not really having any very particularly effective treatments for stroke. So what on earth is it that's driven this? Well, something very important happened around about here, apart from Scotland voting Conservative. And that was that a Welshman sitting as Secretary of State for Health in the UK Department of Health introduced something called the National Health Service, which made healthcare widely accessible, which allowed us to take the public health measures, to treat blood pressure, to advise on smoking reduction, all of these things. And this is the benefit that we see coming from the NHS. And our job, and I think as translational scientists, as basic scientists, we have to understand that at the very best, we can provide the icing on the cake to what these sort of public health measures can achieve. So some sort of semi-closing thoughts. These are some of my favourite. This is advice to the young investigator. Abraham Maslow was a management psychologist in the States. To a man with a hammer, every problem looks like a nail. So not every research question can be answered by the animal model that you already have set up in the lab. Sometimes you might need to change it a little bit, or even, heaven forbid, get a new model. George Santayana, those who cannot remember the past are condemned to repeat it. And finally, my favourite philosopher, Hegel said somewhere that history tends to repeat itself. He forgot to add that the first time is tragedy, The second is farce, and in many ways the history of biomedical research in the UK and beyond these last 20, 30, 40 years has been a tragedy, because we could have done things better and we should have done things better. But now that we know that, let's not make the next 20 or 30 years a farce. Finally, a sort of more detailed thanks to my collaborators. There's a word cloud of people I've published with, and how often. Jeff Donnan, a great mentoring year in Australia. David Howells, a fantastic colleague based in Melbourne. Emily, the best PhD student you could ever hope to have, because A, she worked very hard, and B, she stayed and is remaining productive. And others like Bart van der Vorp in Utrecht, Rustam, Peter, Jonathan, Hannah, who's come up here, who did the MS work, Kieran, another great PhD student. What this word cloud tells you, I think, is that a scientific career requires many, many collaborations, and it's best done if you work with people who are good fun to work with, and it's best done if they work in far-flung places, partly because you get to visit them, but partly because they bring with them their own different traditions and histories and training and knowledge, and that's the sort of thing which makes a career in medical research, for the young people in the room, such a fantastic way to spend your life. And I would like to end by acknowledging the people who have funded me over the years, and thank you for coming along and for your attention. Thank you.
This production is brought to you by the University of Edinburgh.